The Cassini spacecraft was dying. NASA engineers had made the call months earlier: send it plunging into Saturn's atmosphere to burn up like a metal comet. But before it did, they aimed it at something wild: a geyser plume erupting from a tiny frozen moon called Enceladus. Cassini flew straight through the spray, tasting the water, shooting out of cracks in the ice. What it found should have been the biggest story of the century: hydrogen, methane, complex organic molecules. The exact chemical fingerprint you'd expect from hydrothermal vents on an ocean floor, the same kind of vents where life on Earth may have started. And yet, nearly a decade later, we still can't say whether anything is swimming down there. Worse, some scientists are starting to whisper something uncomfortable at conferences: that even if life exists on Enceladus or Europa or buried under the Martian crust, we may never actually find it. Not because it isn't there, but because of a problem so fundamental, so deeply baked into how we do science, that no billion-dollar mission can fully solve it. To understand why, we need to talk about a piece of rock that landed in Antarctica, a lab in Pasadena that nearly ruined a career, and a philosophical trap that's been quietly haunting astrobiology for 30 years. Let me start with something that broke my brain the first time I read it. When I was in undergrad, I took this seminar on origins of life, mostly because it fit my schedule and the professor was rumored to bring donuts. Halfway through the semester, he asked us a question that seemed almost too simple: define life. 30 smart kids, and not one of us could do it without the professor picking our answer apart in under 10 seconds. Reproduction? Mules can't reproduce. Metabolism? Fire has metabolism by some definitions. DNA? That's Earth chauvinism. You're assuming every possible living thing has to look like us. We left that class agitated. Turns out, we weren't alone. NASA has an official working definition: a self-sustaining chemical system capable of Darwinian evolution. But ask 10 astrobiologists, and you'll get 11 opinions. Carl Sagan reportedly hated the NASA definition. Lynn Margulis called the whole exercise futile. And here's the kicker: if you can't define what you're looking for, how do you build an instrument to detect it? This is where things get genuinely uncomfortable, and it's about to get worse. In 1976, NASA landed the Viking probes on Mars. On board were 4 experiments designed to detect microbial life in Martian soil. One of them, the labeled release experiment, run by a scientist named Gilbert Levin, actually returned a positive result. The soil metabolized nutrients. It gave off gas. It behaved, chemically speaking, exactly like the control samples spiked with Earth microbes. Levin went to his grave in 2021, believing he had found life on Mars. NASA officially disagreed, chalking it up to weird Martian chemistry, perchlorates in the soil, doing something no one predicted. And here's the thing that keeps me up at night sometimes: they might both be right, or both be wrong, and we may literally never know, because the Viking landers are dead and the samples are gone. We had our answer, possibly, and we couldn't tell the signal from the noise. Fast forward to 1996. President Clinton walks onto the White House lawn and announces that a meteorite from Mars, a hunk of rock called ALH 84001, found in Antarctica, contains fossilized evidence of microscopic Martian life. Tiny worm-like shapes under an electron microscope. Chemical signatures suggesting biology. It was the most consequential press conference in the history of space science. Within a few years, most of it fell apart. The fossils were probably mineral formations. The chemistry could be explained abiotically. Careers took hits. And this is the pattern that keeps repeating itself in astrobiology: a tantalizing signal, followed by years of debate, followed by the community quietly stepping back from the claim without ever definitively resolving it. Ambiguity is the actual house guest that never leaves. Which brings us to the phosphine problem. And this one is spicy. In 2020, a team led by Jane Greaves announced they detected phosphine gas in the clouds of Venus. On Earth, phosphine is almost exclusively produced by anaerobic life, swamp gunk, penguin guts, that kind of vibe. Finding it on Venus, floating around in the temperate cloud layers, was potentially huge. The paper hit Nature, the news cycle exploded, and then other teams reanalyzed the data. Some found no phosphine at all, others found trace amounts. The debate is still technically ongoing, and depending on which astrochemist you buy a beer for, you'll get wildly different answers. Notice the pattern yet? We keep running into the same wall, and it's not a technology wall, it's an epistemological one. Even when our instruments work perfectly, we can't agree on what the data means, because we don't fully understand the chemistry of these environments in the first place. Now, before I get into the part that genuinely made me lose sleep for a week, I need to tell you about the tardigrades on the moon. In April 2019, an Israeli spacecraft called Bereshit attempted a lunar landing. It failed, slammed into the surface at high speed, and scattered debris across a small crater. What most people didn't know at the time, and what the mission organizers hadn't exactly advertised, was that Bereshit was carrying a payload of thousands of dehydrated tardigrades, little water bears, nearly indestructible microscopic animals that can survive vacuum, radiation, and temperatures near absolute zero. They're probably still up there, probably dead, probably. I bring this up because it's the perfect encapsulation of the second great problem in astrobiology. We are absolutely terrible at not contaminating the places we're trying to study, and this problem is about to eat the entire field alive. Say we send a submarine to Europa. Actually build the thing. Melt through 15 kilometers of ice, deploy an autonomous vehicle into a lightless alien ocean, and it finds something. A wriggling shape, a cellular structure, something that looks alive. How do we know it didn't come from Earth? Every spacecraft we launch carries hitchhikers. NASA sterilizes as aggressively as possible, but bacterial spores are absolute cockroaches. Some strains of Deinococcus can survive radiation levels that would liquefy a human. And there's documented evidence of bacteria surviving on the outside of the International Space Station for years. If we find microbes on Mars, one of the first questions will be, are these Martian or are these Earth microbes we brought with us that adapted? And you can't just look at their DNA, because if life on Mars shares an origin with Earth life, which is entirely possible via a process called panspermia, where meteorite impacts blast microbes between planets, then the DNA might look eerily familiar even if it's genuinely native. But contamination is almost the friendly version of this problem because there's a much darker possibility, and this is the part I promised would keep you up at night. What if life is already all around us, and we've been walking past it for 150 years? This is called the shadow biosphere hypothesis, and it was formalized by physicist Paul Davies and biologist Carol Cleland in the early 2000s. The idea goes like this. Every living thing we've ever studied on Earth uses the same basic biochemistry: DNA, ribosomes, 20 standard amino acids, left-handed molecular chirality. We assume this is because all life shares a common ancestor. But what if it doesn't? What if life started on Earth more than once, and one of those lineages is still here, running on completely different chemistry, invisible to every technique we use? Here's the terrifying part. There's an actual expedition that might have found it and dismissed it. In 2010, a NASA-funded team led by Felisa Wolf-Simon announced they'd discovered a bacterium in California's Mono Lake that could substitute arsenic for phosphorus in its DNA. Arsenic, a poison, something no known life on Earth uses as a structural element. The paper was published in Science. The press conference was massive. And then the biology community absolutely eviscerated it. Follow-up studies couldn't replicate the results. Wolf Simon's career took a serious hit. But here's what haunts me. The critique wasn't that arsenic-based life is impossible. The critique was that this specific microbe wasn't doing what she claimed. The shadow biosphere itself remains an open question. Every microbiologist on Earth uses techniques that assume standard biochemistry. If something existed in a deep hydrothermal vent using different chemistry, we might have literally cultured it, seen it wasn't growing on our standard media, and thrown it in the autoclave. We may have discovered alien life on Earth 1,000 times over and killed it. Now extrapolate that problem to another world. We're sending instruments designed by Earth scientists, calibrated by Earth chemistry, looking for Earth-like biosignatures. If life on Enceladus or Titan is genuinely alien in its biochemistry, our probes might be electronic bloodhounds trained to smell one specific perfume, wandering through a garden of 1,000 different flowers, and coming home empty-nosed. They're a concept in the astrobiology literature called the N should be one problem. Every conclusion we draw about life is based on a sample size of 1, Earth. We don't know how much of what we consider fundamental to life is actually fundamental, and how much is just a weird accident of how it happened here. Water is the universal solvent? Maybe. Or maybe that's like a fish concluding oceans are the only place life can exist. Carbon as the backbone? Probably. But silicon-based life keeps sneaking back into serious papers for a reason. Here's an obscure fact that most space nerds don't even know. In the 1970s, a Soviet scientist named Vyacheslav Krasnopolsky was analyzing data from the Venera probes and noticed atmospheric anomalies on Venus that he privately thought might be biological. He never published the biological interpretation because the political climate in Soviet science made speculative claims professionally dangerous. His notes only came to light after his death. How many other observations like that are sitting in filing cabinets and old hard drives around the world, quietly dismissed because the scientists doing the work didn't want to torch their reputation? Astrobiology has a career risk problem. Claim life too loudly, and you become the next ALH 84001 cautionary tale. Stay too quiet, and real signals get buried. I got to talk once with a planetary scientist at a conference. I won't name them because they'd probably prefer I didn't, who told me flat out that they thought we'd already collected enough data to conclude life exists elsewhere in the solar system, but that we'd never officially confirm it in their lifetime because the standard of evidence has become impossibly high. Extraordinary claims require extraordinary evidence, they said, quoting Sagan. But we've defined extraordinary in a way that basically no realistic mission can achieve. That conversation has been rattling around in my head for years. Think about what it would actually take. You'd need a sample, multiple samples ideally, from multiple locations. You'd need to demonstrate metabolism in real time, not inferred from chemistry. You'd need to rule out contamination 6 ways through Sunday. You'd need reproducibility, which for something on Europa means multiple missions costing multiple billions of dollars each. And then you'd need the entire astrobiology community to agree, which, if the phosphine debate is any indication, is a horse that has already left the barn, been captured by aliens, and been returned as a completely different horse. The Europa Clipper is on its way. Dragonfly is going to Titan. Mars sample return is theoretically happening, though its budget keeps getting slashed like a horror movie victim. These are extraordinary missions, run by brilliant people, and I want them to succeed more than almost anything. But stack the problems on top of each other. We can't define life. We can't clean our spacecraft well enough to rule out contamination. We can't detect biochemistry that doesn't look like ours. We can't get scientists to agree on ambiguous data. We can't fund enough missions to achieve reproducibility. We can't publish tentative results without ending careers. And even if we solve all of that, the most likely discovery, microbes related to Earth life, might not even count as a real answer to the question we're actually asking. The philosophical payoff we all secretly want, the one that would fundamentally shift humanity's understanding of our place in the universe, requires finding life that has no relationship to us whatsoever. Running on chemistry we've never seen, in an environment we barely understand, detected by instruments we haven't figured out how to build, interpreted by a scientific community that can agree on what it's looking at. That's not a mission. That's a miracle. So here's where I've landed after years of thinking about this. There's a real chance we are the only life in this solar system. There's also a real chance we're swimming in it and just can't recognize it. Both possibilities are terrifying in completely different ways. One means we're cosmically lonely. The other means we're cosmically blind. And the most unsettling truth in all of this isn't that we might fail to find life, it's that we might have already found it in a Viking experiment, in a Martian meteorite, in a Venusian cloud, in a lake in California, and simply been unable to recognize what we were holding. The universe might be trying to answer our oldest question, and we just don't speak the language yet. Whether we ever learn to is the real experiment.